Gene editing is powerful, but it looks like the cell still has the final say. Modern genome editing tools differ in what they change and how precise they are. CRISPR-Cas9 brings Cas9 to DNA, cuts it, and lets the cell repair it. It basically breaks DNA and lets the cell fix it. Base editing doesn't cut, it just swaps letters. It converts one base into another. Prime editing goes further. It can rewrite DNA, substitutions, insertions, deletions. No cutting needed. And yes, all of this works in plants. So what's next? Recent March 2026 studies suggest that the next challenge is not simply whether editing works, but why editing efficiency varies. Why does CRISPR sometimes fail? Scientists have found that CRISPR activity is not controlled only by the guide RNA. The cell itself matters. For example, cells contain intronic RNAs that can bind CRISPR components, compete with the guide RNA, or interfere with editing. At the same time, another group is showing that what works in one organism may not work in another, that sequence context matters, and that chromatin accessibility is key. This is leading to a new way of thinking about CRISPR, where the old idea was only CRISPR equals Cas9 plus gRNA. But in the new reality, CRISPR activity should be viewed as depending on Cas9 and gRNA plus genomic context, chromatin state, intracellular RNA environment, and organism-specific biology. Perhaps this helps explain why researchers are increasingly testing multiple guides, multiple genetic backgrounds, and even multiple editing systems in parallel. In wheat, for example, CRISPR-Cas9, base editing, and prime editing were applied to the same target gene. They got multiple functional versions of a known gene, a library of semi-dwarf variants. They obtained a gradient of phenotypes from weaker to stronger semi-dwarf traits. In my opinion, besides solving gene editing efficiency problems, they are also showing an interesting way to optimize a known gene directly in the crop. What do you think?